Okay, this is the reading guide for Chapter 9. I'm going to try to blow through this as quickly as possible, just to keep things hopping. Send me an email if you're having any questions on this. Right away in the beginning, they've got a page that talks about their learning objectives. These are spot on. It's going to take me a second to get there. These are spot on learning objectives right here. You should structure your notes to be able to answer these questions. If you can do these things, I think you will fare pretty well on our quiz. They're going to start talking about coalescence, coalescing genes, and they use a series of trees where they're building up with these gene trees here. So start looking at the figures, look at all the figures. I will probably have figures on the quiz for sure. On the next page, they have a definition for coalescence here. That's important to be able to not just to be able to define the terms, but if you see that word in a sentence, to know what it means. I'll certainly be using these terms, particularly coalescence, in the questions and answers on the quiz. Right down here, we've got a critical meaning that coalescence is not going to show you that that particular allele is the ancestral allele for the population, but that several alleles could have existed side by side as just the one that later became fixed in the population. So make sure you understand that. Down here, they're going to start talking about what affects the time of coalescence. So make sure you write that in your notes. What affects the time of coalescence? List the things that affect the rate of coalescence or the timing of coalescence. They talk here about not being able to reconstruct the complete genealogy of all alleles at a locus in a natural population with complete accuracy. That's true, although ancient DNA studies where we've looked at, in particular, penguins, in these sort of mass graveyards of penguins going back in time, we've been able to look at the rate of fixation of alleles and recover alleles that existed in populations thousands and even 100,000 years ago, but do not exist today, to get an idea of the standing genetic variation and then the rate of fixation. So there's some great work on this. We really know a lot more than we ever used to in terms of rates of fixation and variation in populations. I've mentioned before that one of our goals is to really get the time frame of evolution in our heads. So here's a time frame. This is the coalescence time for human mitochondrial DNA. Take that, open up to the back of your book and the timeline of evolution on there, write it in. See what else was happening around that time in evolutionary history. Know those numbers. This figure here is one of a few that shows you variation in evolutionary rate. This is across alleles for different human genes. There will be a few other figures like this, so pay attention to this variation and what it means. Now we get into the difference between gene trees and species trees. This is something I hope you've at least heard allusions to, and it won't be totally foreign, but this is going to be pretty fundamental for us for the rest of the class here. So make a note to yourself, when do gene trees and species trees show different patterns? Okay, figure that out, answer those questions. I want you to look at this figure and be able to answer, how are these trees different? What factors contribute to make them different? These are the factors that were described, on the last page, right? When I asked you to write down what affects coalescence. So what is it from there that is actually being shown in this figure? So as you're building your knowledge, make sure you're applying the things that you're learning at the beginning of the chapter to answer the questions I'm asking you later in the chapter. This is that part of critical reading where you're taking that knowledge and using it right away. Here we go into methods of molecular phylogenetics. This is what we're going to be doing. This is our application part of this unit. So make a list of the methods and then have a nice space next to or underneath them where you can write down the key assumptions that are different for each type of tree building method. You're going to want to know these because you're going to want to be able to understand the differences between the trees that you're seeing, when you might want to choose a different method over another. That's application for later. For the quiz, though, what you need to know is what are the different methods and what are the assumptions for each one. That's absolutely going to be on the quiz. Okay, here I want to point out morphological versus molecular homoplazy. How are they different? That's going to be an important one. I will talk to you more about these different methods, so I'm not going to spend your time on it now. You can see I've highlighted a couple of things in the text that I thought were important. Okay, here we're going to get into experimental phylogeny, and this is just fantastic. Hillis at UT did an incredible study to really be able to understand, to really be able to test our methods of phylogenetics, and so to compare them, right, in a known system. So I want you to be able to answer, what's the take-home point of Hillis's experiment? What did he find? And one of the things they're going to talk about here in this section, and they're going to show in a figure here, is the increasing complexity, the rapidly increasing complexity in terms of number of possible trees for what turns out to be actually quite a small number of taxa. So here, unrooted trees. That's a lot. I can't even read that on my screen. How many, what, millions, billions, what are we in, trillions here of trees, possible trees, for just 13 taxa, which means that the computational load to be able to test every single possible tree, impossible, not possible. So, right, these methods are seriously computationally intense, and they need to have a model, right? You can't test every single tree, and you need a model anyway. So keep moving. 4 cases in molecular phylogenetics. Basically, skim a little bit through these, but know what are the take-home points, right? So origin of tetrapods, and I sort of have the take-home point here. So know what this take-home point means. Fossils make a prediction, and the DNA supported it. So what was the prediction, and how did the fossils support it? The human example is one I'll possibly quiz you on. You remember we already have the coalescence time for mitochondrial DNA, so we're picking up some human information. In this section, I'm thinking, what is the evidence that humans originated in Africa in terms of the divergence pattern? What type of evidence did the divergence pattern show? And then also, what type of data, what types of data were used that also supported the origination in Africa? Here's an example about Darwin's finches coming up here, which sort of pulls you out to among species rather than within species, which is a nice one. Everybody loves Darwin's finches. They're going to talk about HIV again, and I just highlighted that this was super duper cool, that there are 3 lineages of HIV, right? So HIV-1 arose 3 different times here in yellow, with, within humans coming from chimps. So 3 different independent origins of HIV-1. And what they point out on the next page that got me so excited was that all 3 lineages had acquired the same mutation encoding the same new amino acid in the same position in the same protein. Wow, I really thought that was amazing. Okay, so natural selection versus neutral evolution. This is great. This is where we start talking about evolutionary mechanisms other than selection. I know we haven't spent a huge amount of time on selection yet, a little bit in this last unit, but neutral evolution is really important for phylogenetics. So make a list here. What are the ways that sequence changes can escape selection? Should at least have 2 or 3 different ways. And then I want to point out something that they don't explain very well here, but they do later in the summary. And that is that the key difference between selection and neutral evolution is that neutral evolution, that rate is regardless of population size. We know that selection is going to be faster in smaller or larger populations? That's a question you should be able to answer from the beginning of the chapter. Here, neutral evolution regardless of population size. Then I just want to point out here, they've got a phenomenal label on this graph. This label tells you exactly what you need to know from this figure. So read that and strive for the rest of your life to write labels as good as that. Okay, here is a question to be able to answer. This says, this variation, shown above in this figure, right, is a prediction of the neutral theory of evolution. How is that a prediction? I want you to be able to explain that. Under molecular clock, they have this sentence here, to measure the divergence of species separated by hundreds of millions of years, a slow evolving segment of DNA will provide greater accuracy than a fast evolving one. Why? Why would you want to use something that evolves slowly when you're looking for deep divergences? If you were to use a fast evolving one, what kinds of problems might you encounter? This is something you should have picked up in the earlier reading. So again, we're trying to apply the knowledge as soon as we start getting it. And then they're going to talk about a relaxed molecular clock. Why would you want to use a relaxed molecular clock? They've been trying to nail home this idea this entire chapter. So why would some sites have a faster rate of evolution than others? Why would some branches have a faster rate of evolution? Be able to put that into context. Think about the figures in the text and which of them show evidence that a relaxed molecular clock is justified. They'll talk about using fossil calibrations to improve the estimates for molecular clocks. We'll talk about that more in class. And then they give you 2 results of this study. Okay, mostly what I want to focus in on is some of the more broad level things here. So now they're going to talk about how do you test selection. So down here, this is the null hypothesis for when you're testing for evidence of selection at the bottom of the page there. And then they're going to give you examples of comparing different taxa to identify mutations, where you can be comparing different individuals within a species to a closely related species, or you can just be comparing multiple closely related species. So here again, this critical reading, I want you to be thinking like this. If I pose this kind of question, when you make a phylogenetic tree to investigate the species relationships, you always do a test for selection. If you see evidence for selection, like one of the tests that they're going to describe here, usually throw out that sequence. So generally you would throw out that entire locus. So I sequenced, say, 13 loci to investigate the species relationships for Hawaiian honeycreepers. I only published on 12. I threw one out because I saw evidence of selection. Why would I throw that out? Think about gene trees versus species trees and what you would expect to see there. Be able to answer that question. That's easily something I could put on a quiz to see that you're really applying the knowledge. Okay, this is a great figure here, one that I would like to be able to put on a quiz, for instance, to see if you can correctly identify which mechanism would be most likely to be acting in each of these scenarios. So purifying, neutral, positive selection. So if I were to just put one of these on, could you identify what type of selection it is? Or if I were to say, draw me what it would look like if you had positive selection, could you do that? This part starts to get a little bit confusing, so we're going to apply that later. Basically, what I want you to pay attention to is here in this table. They start talking about all these different tests for selection. Look at these columns. So look at the scope of comparison. We've got between species, and within a population. We can look for evidence of positive selection, and we have one test for purifying selection. There are several tests for time frames greater than 1 million years. Also, a test that you can use for less than 200,000 and less than 30,000 years. So you're going to use different tests for different questions at different time frames and within or among species, right? So I just want you to know sort of these broad principles in terms of time frame for evolution for tests. Here, this is that summary that I was telling you before, where they really explain neutral evolution really nicely in here. So read this key concept here. And actually, whoever wrote the learning objectives and the concepts and summaries, they did, I think, an even better job than whoever wrote the chapter, although this is a really good chapter. They're going to give you a couple of examples here in deciphering the genome. These are nice examples, very interesting, looking at humans and selection and comparative genomics. I'm not going to test you on those, but I really enjoyed reading them. Make sure you take a look at the multiple choice questions. Those are a good, very basic question for yourself. I'll have probably harder questions on the quiz, so if you can't answer these, then you're going to want to make sure you reread the chapter a couple times. All right, good luck.